Welcome to OncoLife interview session. It's my pleasure to introduce our guest today, Dr. Nicolas Girard, Professor of the Respiratory Medicine at Versailles Saint Quentin University and lead investigator of the LUMINOSITY trial. You are welcome.
Thank you.
Thank you very much for joining us today. As you know, antibody-drug conjugates represent a significant breakthrough in oncology, offering targeted treatment, especially for lung cancer. Dr. Girard's work has been at the forefront of this cutting-edge research, particularly in targeting protein biomarkers. Can you share some information about your background, your research, and your professional journey?
So I'm a respiratory physician by training, now thoracic oncologist at Institut Curie in Paris. So mostly dealing with patients with lung cancer, non-small cell lung cancer, and interested in immunotherapy, targeted agents. So obviously the new era that is now occurring with the antibody-drug conjugate is something of high interest, not only for the treatment of patients, but also for the understanding of the biology behind the efficacy and safety of these new agents.
And how does your research at Institut Curie integrate precision medicine principles, particularly in targeting overexpressed protein biomarkers in solid tumors?
Well, that's a very good question. We are now moving from a dogma of molecular alterations based on the activation of oncogenes, activating mutations, gene fusions, predicting the oncogene addiction in tumors, towards a paradigm of finding the biomarkers to help with the entry of cytotoxic agents through the combination with antibodies. So it's a quite different paradigm in terms of biomarker, moving from NGS based on DNA, RNA alterations to a more functional way to see the biomarkers with the expression of these targets as a surface of cancer cells. This requires obviously different techniques, mostly immunohistochemistry, but it also means that this expression may change over time and along with the exposure to anti-cancer agents and along with the disease evolution. So this is something very important because these targets may change from the time of diagnosis to more advanced disease.
Could you elaborate on the potential of the antibody-drug conjugates like telotuzumab and ventodine in overcoming the challenge of treating solid tumors such as lung cancer and colorectal cancer?
Yes, so Telizovy is an antibody-drug conjugate targeting c-Met, which is a target that we see. This is an oncogene that is a target and that we see at the cell surface of cancer cells in lung cancer and other solid cancers. Clearly, Telizovy has demonstrated anti-tumor efficacy from this LUMINOSITY phase 2 study. So in patients with advanced refractory non-small cell lung cancer after the failure of first-line treatment with chemotherapy and immunotherapy. In this phase 2 study, the target population where we see the highest efficacy are patients with non-squamous cell carcinoma and overexpression of c-Met without other oncogene alterations. At the end, in this subset of patients, we see a response rate of about 30%, even higher in c-Met high patients. So there is a kind of correlation between the efficacy and the magnitude of expression in terms of percentage of positive cells in the biopsy, a cutoff that was determined after looking at phase 1 and first-in-man data. So this is a really strong cutoff correlating with response rate. At the end, we have a signal in terms of progression-free survival and overall survival that seems to be higher than the standard of care, which is docetaxel in the second-line setting for non-small cell lung cancer. So we have here data that support the anti-tumor efficacy of Telisovy in c-Met overexpressing tumors. And now we have a phase 3 trial, which is TeliMet, comparing Telisovy to docetaxel in those patients with non-squamous cell carcinoma, EGFR wild type, and c-Met overexpression. So it means that this requires to look at c-Met overexpression before starting the trial. It's good to have a rebiopsy. If c-Met is negative at baseline, maybe it will become positive after previous exposure to chemotherapy or immunotherapy. So this is the first point, and pre-screening can be done quite easily because this is the SP44 Ventana antibody, which is widely available. This is what we do here at Institut Curie. Enrollment of the patient in the clinical trial if the patient is eligible, and this obviously will lead to look at overall survival, progression-free survival, benefit of Telisovy versus docetaxel with a high level of evidence.
And what makes c-Met and SEZ6 particularly compelling target for ADCs, and how do you anticipate these targets will impact the treatment paradigm in lung cancer and other solid tumors?
Well, there are other targets in lung cancer for ADCs, HER2 mutations, 1 to 2% of non-small cell lung cancer. We have some data from the phase 2 trial Destiny Lung 01 with trastuzumab deruxtecan in those patients. Maybe HER2 overexpression, which is another subset of patients, could be targeted as well by this drug. And then there are obviously other ADCs targeting MET, such as Teliso-A, such as Mytix 011, which are under investigation in phase 1 studies. Other targets are TROP2, with sacituzumab govitecan, negative trial. Datopotamab deruxtecan demonstrated some kind of benefit versus docetaxel in a randomized study in the second-line setting after chemo and immunotherapy. In this TROPION-Lung01 trial, we see a benefit in terms of response rate, but of more importance in terms of progression-free survival and overall survival in non-squamous cell carcinomas, which is a subset of the ITT population. There was no selection based on TROP2 expression in this trial, but of high interest is that if we revisit the data in this trial, so datopotamab deruxtecan in the second-line setting, no selection in the clinical trial based on TROP2 expression, but if we retrospectively look at the data, especially looking at the TROP2 expression with a kind of artificial intelligence algorithm, which is called the QCS, the quantitative expression of TROP2 expression, especially the membrana expression, well, we have some correlation. And this was shown at the last WCLC Congress. And at the end, this QCS scoring, which is a way to quantify immunohistochemistry, probably in a better way than the human eye of the pathologist, could be a way to define cutoff of expression for TROP2, predicting the efficacy of datopotamab deruxtecan. So this new way to perform immunohistochemistry and kind of digitalization of immunohistochemistry biomarker could be a way to define targets for ADC-based strategies.
And what are the key takeaway from the phase 2 LUMINOSITY trial regarding the efficacy and safety of the Telisov in patients with the CMAT overexpressing EGFR wild type of the non-small cell lung cancer?
Well, this is what I described, the overall response rate of 30%, which is higher than that of docetaxel, usually 15% maximum. So we have here a strong rationale to move to the phase 3 with this response rate, PFS 7 months, overall survival 14 months, median. So it's quite high as compared to what we have. We also reported the patient-reported outcomes from LUMINOSITY. Very interesting analysis because we see that we have a more prolonged time to deterioration of quality of life and symptoms in patients with response. This is quite expected, but also in patients with stable disease, meaning that stable disease in this advanced refractory setting is meaningful for the patients in terms of quality of life. We also see improvements in cough and chest pain, which are key cancer-related symptoms. What about the safety of Telisov? Well, this is an ADC, so we have side effects that are related to MET inhibition, mostly edema, quite rare, and side effects related to the payload, meaning the cytotoxic chemotherapy attached to the antibody. And with vedotin, we have neuropathy, peripheral neuropathy, which is a kind of cumulative side effect, so it's only seen in quite long-term responders. And we also see that not only in the report of the side effects, but also in the patient-reported outcome. Hopefully, the grade 3-4 neuropathy is quite rare, and at the end, it's manageable with prevention. But again, it's a quite specific side effect with this ADC. And this is something that we have to learn as clinicians. At the end, the safety profile of ADCs overall is quite different than that of chemotherapy. We have more frequent ILDs, interstitial lung disease, more frequent stomatitis, ocular toxicities, in addition to nausea, vomiting, in addition to hematological side effects. So at the end, we need to learn. And this is what we do at Institut Curie with this concept of ADC clinic, meaning identifying who are the specialized specialists for each side effect, and learning all together on how to manage those side effects. So at the end, this is really something that helps with the proactive management of these side effects, meaning I'm not waiting grade 2 before starting a treatment because there is a high risk to move to grade 3 and grade 4. I do prophylaxis for these side effects. I have a close monitoring not only with specialized physicians, but also with dedicated nurses and so on. So this is something that we need to learn. We did that when immune checkpoint inhibitors came to the clinic, and we have to do the same with ADCs.
Can you discuss the significance of PRO in LUMINOSITY trial, particularly in assessing quality of life and symptom management in lung cancer patients treated with Telisov?
So this is what I reported, the patient-reported outcomes. It's something very important, especially in the phase 2 study, to understand what is meaningful for the patients. Here we have a new mechanism of action for this drug. We need to learn whether the side effects that are reported are actually impacting the quality of life, to what extent. And we also need to learn what is the relationship between response, radiologic response, and the actual benefit for the patient. So it's really important to me to include this PRO approach for the assessment of new agents, especially in the setting of phase 2 trials. It adds to the value, to the assessment of the value of the compound, not only for the healthcare authorities, but also for clinicians and patients.
Are there any plan to explore the telisovir in combination with other therapies such as immune checkpoint inhibitors and targeted therapies to enhance treatment efficacy in the especially lung cancer?
Well, that's a very good question. ADCs may be combined with immune checkpoint inhibitors. There are many trials ongoing with the TROP2 inhibitors, sacituzumab and datopotamab deruxtecan. We know that this is a combination that is safe. We do not have increase of the side effects or synergy in the side effects of these compounds. It's an opportunity in lung cancer to move from late line to first line, where the standard of care is based on immune checkpoint inhibitors. So obviously this is something to explore. And then the question is how to select the patient population in terms of biomarkers, not only for IO, but which is the biomarker being PD-L1 expression, but also for the ADC.
What are the next steps in your research on ADC in precision medicine? Are there any upcoming trail for novel target that you are particularly excited about?
No, there are many targets, obviously. The question will be how these targets are co-expressed or not. If we have a panel of targets and a panel of ADCs, what will be the treatment sequence? Which target has to be prioritized? And maybe this is a matter of quantification at the end, and quantification with this QCS and digitalization of immunohistochemistry slides is probably a very promising approach in my sense. But I agree with you, it's a very different setting than the molecular alteration, which are mutually exclusive one to another. Here we are clearly dealing with multiple biomarkers which may be co-expressed.
Thank you. This is my last question. How do you envision the role of ADC evolving in the broader landscape of the oncology treatment? What breakthrough do you anticipate in the next 5 or 10 years that could significantly alter patient outcomes?
Well, that's a very good question. We are moving towards combination and escalation with the combination of ADCs, with chemo, with immune checkpoint inhibitors. The question is who is benefiting, who requires such escalation, and we probably need to have a better stratification of the prognosis of patients to know who are the patients who require such intensification and who are the patients who may not benefit from these combinations.
Thank you very much for all this important information and sharing your expertise and insights. It's a pleasure for us to have you today.
Thank you.